Okay, I am excited to be here. Thank you very much for the last event of this seminar series. So before I jump into the main part of my talk, a little bit of background information. So in addition to my position at DIME, I do hold a part-time research and teaching faculty position as shown on the screen. And I also have some financial disclosures as listed as well. The disclosure regarding Philips is particularly relevant today, as I will be presenting some data from a Philips sponsored study, which was undertaken while I was an employee there a couple of years ago. All right, so brief outline of my talk today. I'll spend a few minutes at the beginning introducing you to the Digital Medicine Society, or DIME, as I'm aware that many of you here today may not be familiar with who we are and what we do. I'll then provide an overview of the V3 framework and lead into two examples specifically to the field of sleep research and sleep medicine, which illustrate the application of the V3 framework. And I'll wrap up by pointing you towards some additional V3 related resources which might be helpful in your own work. So as Anna just mentioned, DIME is a registered 501c3 nonprofit organization, and we are dedicated to advancing the ethical, effective, equitable, and safe use of digital medicine. We are an organization for professionals and sitting at the intersection of the healthcare and technology fields. The Society was launched as a nonprofit in mid-2019, and at that time, we identified 12 industry sponsors who were collecting digital endpoints in their clinical trials. So by industry sponsors, I mean biopharmaceutical companies or medical device manufacturers who were using digital tools to collect their endpoints in their clinical trials used for development of new drugs, devices, or biologics. And in total, back in 2019, we identified 34 unique endpoints captured by digital measurement tools. As of May of this year, so just under 2 months ago now, the number of sponsors has increased to 107, in total collecting 396 unique digital endpoints, 46 of which are related to sleep. So that's enormous growth in a very short time. So an almost tenfold increase in the number of industry sponsors working in this space in a period of under 4 years. And that is, undoubtedly driven by the fact that digital measurement tools do offer several advantages over traditional methods of data collection in clinical trials. So, for example, a non-exhaustive list here, collection of data outside of the clinical or research setting is likely to better reflect the lived experience of patients and participants enrolled as in trials. And of course, by using digital tools, not only for data capture, but also for supporting recruitment, things like e-consent and patient engagement, those types of tools and the opportunity for decentralized data collection is likely to improve accessibility, recruitment and retention, and ultimately, hopefully, lead to a sample that is a better reflection of the overall patient population of interest. Using digital data collection tools provides the opportunity for objective data, which is a nice complement to self-reported, patient-reported outcomes of things like functional status and health-related quality of life. And high-resolution data. So by that, I mean either data collected continuously throughout a participant's enrollment or data collected at very frequent intervals throughout the enrollment period. If modeled individually, that will increase the statistical power of a trial and, of course, bring the required sample size down. So all of this adds up to a lot of opportunities for improving the efficiency of implementing trials, which traditionally is a time-consuming and a very expensive process. However, all of these advantages do come with some challenges as well. One of which, a biggie, is that there are just so many of these digital measurement tools now available, especially in the field of sleep. So it can be really challenging for clinical researchers, sponsors, clinicians to identify a tool or a group of tools that may be appropriate for a particular clinical trial. And that is what led us to development of the V3 framework, which was published by DIME in NPJ Digital Medicine in 2020. So V3 is a generic assessment framework, and it guides the reader or the user through the necessary steps to determine whether a particular digital measurement tool is fit for purpose. And we use that term fit for purpose intentionally, recognizing that one particular tool may be a fantastic resource for one clinical trial. It doesn't necessarily mean that it is perfect for another clinical trial, which might have a different design and a different patient population. So, I mean, one thing I hear pretty frequently is statements like, oh, this tool is great because it's been validated. And in my head, I immediately start thinking, but, you know, validated for what? To do what? In what trial? In what patients? So V3 is a resource which anyone can access and use if they are either developing digital measurement tools or assessing them for use, either in a clinical research setting or a clinical care setting as well. So moving into an overview of the framework itself. Before I get into it, let me just quickly define some terminology. So the paper published in 2020 refers to biometric monitoring technologies, or biomets for short. The definition of a biomet is on the screen. I'm not going to read it out word for word, but there are 4 key characteristics of the types of tools that we're talking about here. So the first is that these are tools that are connected, meaning that there is a digital method of data transfer. Oftentimes these days, the data transfer is wireless. So it might go from the tool itself to an app and up into the cloud, but it doesn't necessarily have to be wireless. These tools are mobile, meaning that they are capable of collecting data outside of the clinic or research setting without the support of specialty staff. And by definition, these are tools that use a combination of sensors and algorithms. So sensors that capture some kind of physical construct like light, temperature, pressure, acceleration. And then an algorithm or series of algorithms uses those sensor-generated signals to estimate a measure of behavioral and or physiological function that is somehow applicable in medicine. So these are the tools that we're focusing on. For this sleep-focused audience, the simplest example is actigraphy, but a biometric monitoring technology can be something far more complex with lots of different sensors and input signals with multiple series of algorithms as well. So I'll walk through now the 3 V's of V3. The first is verification. I'm sure this is a process that's familiar to many of you. Verification evaluates and demonstrates the performance of the actual sensor, which is contained within the form factor of the tool itself. So it might be a wearable, it might be an environmental sensor, like something that sits next to the bed or a mattress pad or something like that. It can also be an implantable or an ingestible sensor. But regardless, the verification process focuses on the quality of the sensor or sensors. Next up, we have analytical validation, and this evaluates the performance of the algorithm or algorithms that takes those sensor-generated data and converts it into an outcome or a clinical trial endpoint, which has some kind of clinical meaning. And then finally, clinical validation. This is a question around whether or not the outcome or endpoint is clinically meaningful. So whether it identifies, measures, or predicts a meaningful clinical state within a context of use, which includes a specified patient population. So the term V&V, verification and validation, this is a term or a concept that's been around for many, many, many years, coming out of the engineering literature specifically. What V3 does, and actually we were not the first to do this, is take the validation piece and split it into 2 components, because analytical validation focused on algorithms is specific to digital tools, whereas clinical validation and verification are concepts that are applicable across the board. So V3 processes are multidisciplinary. So verification, that is something that happens entirely at the bench. It does not involve human participants, and it is typically the responsibility of an engineer or a team of engineers. Human participants come into the picture for both of the validation components, so analytical and clinical. Clinical validation is typically the responsibility of clinical researchers, clinicians, and sponsors, whereas the middle part of that Venn diagram, analytical validation, that is best undertaken as a collaboration between engineers and clinical researchers, investigators, and clinicians. Importantly, there is no expectation that one particular researcher or lab or team is undertaking every component here. So typically, and as emphasized on this slide, it is a multidisciplinary series of components best undertaken by a collaboration. All right, let me now walk through a very high-level, simple example of the 3 V's, and then we'll move into some sleep-focused examples too. So verification, does the sensor generate data that are good enough to then feed into the algorithm, which is then the next step? The algorithm is then the focus of analytical validation, which would answer a question like, does the algorithm measure what it says it's measuring? And then finally, clinical validation is the endpoint that has been selected for use in a trial or for in some kind of clinical care application, clinically meaningful within a stated context of use. So let's say, just as an example, we have a simple actigraphy tool. So this is a wrist-worn tool that contains a 3-axis accelerometer. Now, by definition, of course, an accelerometer samples acceleration in units of meters per second squared, or G. So to complete the verification process, we need to identify a ground truth for acceleration, which in this example would be something like a shake table, like the one you see on the screen. There are plenty of these types of things available. An engineer or a team of engineers would then place the accelerometer sensors on the shake table and make a comparison between what is detected by the sensor versus the known acceleration, to assess whether or not the accelerometer data are accurate, precise, uniform, and consistent. So throughout today's talk, I'm going to highlight 4 key questions that will come up along the way. And then later on, I'll circle back and we'll go into them in more depth. But for now, question 1, what if the raw data are compressed into activity counts? This is pretty common. There are a lot of actigraphy devices that do this, but it's not limited to just actigraphy. So I'll come back to that question in a few minutes. Moving on now to analytical validation. In our actigraphy example, an algorithm is applied to the accelerometry signal, and that might generate, for example, an estimate of sleep efficiency. So when we're considering analytical validation, we need to identify an appropriate reference standard for sleep efficiency. Now, obviously, we have one that is very obvious, and that is an in-lab PSG. So an analytical validation study might, for example, address the question, what is the epoch level agreement of sleep and wake between the actigraphy tool and the manual scoring of PSG? And then finally, clinical validation. In our hypothetical example, say we have a researcher who is considering using this tool in a clinical trial of patients with mild cognitive impairment. They will need to identify the fact that, or compile the evidence that the endpoint, so for example, change in sleep efficiency, predicts a meaningful clinical experience. So they might be interested in the question, does sleep efficiency predict the onset of dementia in older adult patients, for example? So verification is focused on the quality of the sensors. Analytical validation is focused on the quality of the algorithms. And finally, clinical validation is focused on the clinical relevance or meaningfulness of the endpoint. So the 3 processes that I've just walked through are modular, so they can be thought of as separate components. So for example, if there is a change to the hardware or the firmware of a digital measurement tool which impacts the sensors, there would be a need at that point to go back and redo the verification process. But having done that, assuming your verification study meets whatever pre-specified performance criteria you laid out in advance, there would be no need to then progress further and redo all of the analytical validation studies. And similarly, if there have been changes to the software that impact the algorithm, there would be a need to repeat the analytical validation process. But if the sensors haven't changed, there's no need to go back and redo verification. So we can think of these three processes as being related and in sequence, but separate. All right, I'll close out this section and move into some sleep-focused examples. But just to emphasize before I do that V3 is just one step with three components. And it's the first step of a much more comprehensive evaluation. So, for example, it is also important to think about things like security, data rights and governance, human factors and usability of a tool, and of course, the economic feasibility of implementing a particular tool that you might have in mind. Okay, so that is a quick summary of V3, and I'm now going to move into a couple of examples specific to sleep. First example on the left is a theoretical application, and it walks through all three components of V3 using wearable dosimeters and light loggers as an example. Everything I'm about to describe in the upcoming slides comes from this 2022 paper published in Digital Health. First author was Manuel Spitzan, who invited me to speak today. And this review was put together a couple of years ago under the DIME Research Committee at that time. So when we're thinking about wearable dosimeters and light loggers, all of us, of course, are exposed to various artificial and natural light sources throughout our days that are defined in time, space, and the spectrum of the light. And all of that combined adds up to our ocular light exposure. And what we're actually interested in from a biological point of view is the light that reaches the retina. However, we're not able to measure that directly. So instead, we use light sensors, ideally placed somewhere near the cornea, which sample light exposure and are measured over time and combined into various summary metrics. They might be simple, such as the maximum light exposure within a given day, or they might be much more complex than that. And we then use those summary metrics to determine which have an impact on various chronobiological health outcomes. So let's think about the three components here. So verification, compared to a reference standard, so that's the first thing, is to identify a ground truth for light, something like a lux meter. Is the light sensor itself accurate, precise, consistent, and uniform? And of course, because we're thinking about light, we need to think about things like the spectral properties of the light sensor, the potential for an ultraviolet or infrared response within the visible wavelength, the directional response, especially depending on where exactly the sensor is placed. So it might be on a watch, or it might be on glasses. It might be an environmental sensor that's not connected to the body. And of course, there may be the potential for environmental considerations, such as the performance of the light sensor under different temperature or humidity considerations too. So all of that is an engineering-focused assessment of the quality of the light sensors. Moving into analytical validation now. Analytical validation focuses on the algorithm that takes that light sensor data and estimates personalized light exposure of some kind. So when used by participants under controlled settings, so by now the light sensor is contained within the form factor of the tool itself. So it might be a watch, as I mentioned before, it might be glasses. Compared to that reference standard, typically in a lab setting, does the tool itself, the light logger, capture what we believe to be the true light exposure? And then finally, clinical validation on the right-hand side of the screen. This is a much bigger picture question, and it evaluates the extent to which the summary metrics of light exposure over time for a given patient or participant predict some kind of important health outcome of interest. So verification, to reiterate, assesses the quality of the light sensors. Analytical validation assesses the quality of the algorithm that converts the spectral data into some kind of clinically relevant outcome. And clinical validation compiles the evidence that the outcome chosen that is based on light exposure is clinically relevant for a particular use case and for a particular patient population. So that is a very, very high level summary. There is much more in-depth information contained in the 2022 digital health paper with the reference on the screen here. But I'll move now into a second example. This is a real example from the peer-reviewed literature, whereas what I just described for light loggers was a theoretical application. And in this second example, I'm going to hone in on the middle component of V3, so analytical validation only. In my experience, and certainly when I talk to several of my colleagues, it's the same for them as well. Most people are pretty savvy with the verification and clinical validation pieces, but analytical validation, that middle component of V3, which is specific to digital tools, this appears to be the process that generates the most confusion and questions. So I'm going to do a bit of a deeper dive just on that one process, using cardiorespiratory sleep staging as an example. I mentioned up front that I will be presenting data from a Philips-sponsored study, and this is the one. So we are now moving into an example that comes from the clinical sleep literature. And the problem that we were tackling here was the absence of PSG signals in home sleep tests. So home sleep apnea tests are now very commonly deployed as a diagnostic test for sleep apnea. But the absence of PSG signals, so no EEG, EOG, or EMG, means that we do not have a method for calculating total sleep time. And therefore, if we're using a home sleep test, we can't calculate the AHI, or the apnea-hypopnea index. The AHI, for better or worse, is the most widely accepted kind of gold standard for assessing the severity of sleep apnea. And that is the total number of apneas and hypopneas divided by the PSG-determined total sleep time. Without those PSG signals, we instead need to use a different denominator and calculate the REI instead. So that's the respiratory event index. Most commonly, the denominator for the REI is the recording time. There are other alternatives, but this is by far the simplest and it's by far the most common. The REI almost always underestimates the AHI, because total sleep time is almost always less than recording time. We never see patients that sleep for the entire duration of a home sleep test recording. And so what this means is that home sleep tests have reduced diagnostic sensitivity compared to the gold standard methodology of PSG. So the digital product that we are assessing in this study is a cardiorespiratory sleep staging algorithm, so CRESS for short, which estimates simplified sleep staging using an airflow and heart rate signals at minimum, but also a single respiratory effort signal if available. And our objectives in this study, summarized in the JCSM paper 2021, was firstly to undertake an epoch-level comparison of the sleep staging algorithm compared to manual scoring from a PSG. And next, to determine the impact on the accuracy of the respiratory event index compared to the gold standard AHI. Question number 2, which I will come back to, what if the algorithm isn't open source? And I'll kind of preemptively answer this question right away by letting you know that this particular algorithm that I'm providing this example for is a proprietary algorithm, so it is not currently open source. Okay, so going into the methods here, if we have a full PSG that includes the neurological signals as well as all of the cardiorespiratory signals, including airflow, oxygen saturation, etc., we can tally up the respiratory events, divide it by PSG total sleep time, and calculate the AHI. And that is what determines the severity of sleep apnea. If we limit the montage down to just cardiorespiratory signals, we can then calculate the respiratory event index. So total number of events divided by recording time as the traditional and most common home sleep apnea test severity index. Alternatively, and what we are proposing in this particular paper, is to instead calculate the REI based on algorithm determined total sleep time. So total number of events divided by the algorithm total sleep time to give the CRESS REI as our proposed home sleep apnea test severity index. Now, what we did not do as part of this analysis was make a direct comparison between the two REI values, because the gray REI, the traditional one based on recording time, is not what we consider the gold standard for determining the severity of sleep apnea. Instead, we compare each of the individual REIs back to the gold standard, the AHI, and calculate the intra-class correlation coefficients, and then make a comparison of those two correlation coefficients. And that will tell us about the accuracy, the relative accuracy of each of the REIs that we have generated. So to do this, we accessed de-identified PSGs from the National Sleep Research Resource at sleepdata.org. We identified 296 PSGs from the MESA cohort study and another 296 from the Sleep Heart Health study. So in addition to being able to access from sleepdata.org the raw PSG signals, we're also able to access the PSG annotation files. And the annotation files compile the original scoring that was done by the sleep technologists in the original MESA and Sleep Heart Health studies when they were undertaken some years ago. So we got the PSGs and the annotation files and imported them into our software and combined all of the light sleep stages. So for MESA, we took all of the N1 and N2 epochs, combined them and relabeled them all as light sleep. For Sleep Heart Health, which was a study done now several years ago, which used the older R&K scoring criteria, we took all of the stage 1 and stage 2 epochs, combined those and relabeled them as light sleep. After doing that, we created a home sleep apnea test montage by taking out all of the signals other than airflow, heart rate, and respiratory effort. And then using those three signals, we deployed the cardiorespiratory sleep staging algorithm, which generated an estimate of simplified sleep staging. So the reason that we chose the MESA and Sleep Heart Health cohort studies, there were actually many reasons, but the two key reasons was one, these were PSGs that were collected in the home setting. And two, more importantly, they used different airflow and heart rate signals. So MESA, for example, used a nasal pressure signal for airflow and a PPG signal for heart rate, whereas Sleep Heart Health used nasal thermistry for airflow and an ECG signal for heart rate. So we specifically wanted to test different input signals when we looked at algorithm performance. So here are the primary results. This is a confusion matrix done at the epoch level. So starting with the left-hand column under wake, these were all of the epochs across the 296 PSGs from MESA that were identified as wakefulness by the original sleep techs who were involved in the MESA study. And as you can see in that very first green cell, the sleep staging algorithm correctly identified 60,000 or just under 60,000 of those epochs, meaning that the algorithm correctly identified 76% of all of the wake epochs. And as we move down that diagonal line with the green cells, the algorithm correctly identified 85% of the REM epochs, 85% of the light sleep epochs, so that's N1 and N2 combined, and 50% of the deep sleep epochs. So question number 3, which I will circle back to, is how granular should my analysis be? So by granular, what I'm referring to here is that this analysis, the confusion matrix, is based on individual epochs rather than summary metrics like total sleep time or sleep efficiency, for example. Our second objective was to look at the impact on the cardiorespiratory sleep staging algorithm in terms of the accuracy of the respiratory event index. So this figure is a little busy. I'll just walk through each component individually. So first, looking at the orange line, the diamond in the middle here, that is the intra-class correlation coefficient between the traditional REI, so the respiratory event index calculated using recording time, compared to the AHI, calculated using the manually scored PSG determined total sleep time. So the correlation coefficient there is 0.85. Now the width of the orange line is the 95% confidence interval, and you can see that it's fairly wide. At the lower bound, we have 0.51 up to the upper bound of 0.93. The blue data that you're seeing towards the right of the figure, in the middle here, the diamond is the intra-class correlation coefficient between the respiratory event index, which instead uses the algorithm-determined total sleep time, compared with the gold standard AHI based on PSG manual scoring. So it's a little hard to see, but I'll put some shading behind it so that it is a little clearer that what we're looking at here is that the lower bound of the blue confidence interval is higher than the upper bound of the orange confidence interval. So the lower bound of the blue is 0.94 compared to the upper bound of the orange, which is 0.93, meaning that the respiratory event index based on the algorithm determined total sleep time is a significantly better estimate of the AHI than the respiratory event index based on recording time. Now, all of the data at the top here are generated when the algorithm uses only two input signals, so airflow and heart rate. If we now allow the algorithm to also use the thoracic respiratory effort signal, we see no change to the orange line because nothing has changed. That additional signal doesn't make any difference to the recording time. But what we do see is a change in the blue data, as again, I think is clearer when I put some shading behind it. So now the correlation, the blue diamond, the correlation between the novel or proposed REI versus the gold standard REI is 0.97, I'm sorry, and the confidence interval is 0.96 to 0.98. So there is a larger gap there, meaning that using that additional signal, the respiratory effort signal, results in a further improvement of the accuracy of the REI when the algorithm generates the total sleep time denominator compared to the gold standard AHI. So that was a lot to digest in just a couple of slides, but what I've summarized there are two separate components of analytical validation. The first is a validation of the most granular data. So in this scenario, we're talking about individual epochs. And once that was done, we then move into a comparison of the summary metrics. So in this case, the summary metrics being the AHI versus the REI with and without the proposed sleep staging algorithm. Now, of course, all of that only falls into one component of V3. So it was also the responsibility of our team to put together some evidence that confirming that all three signals were collected with verified platforms. And thankfully, all of that information is available through the National Sleep Research Resource. And also at the other end, to compile the evidence that the AHI is a clinically relevant metric for determining the severity of sleep apnea. Now, there is obviously some debate around the AHI. It's not really optimal, I guess, is a good way of summarizing it. That's a topic for another day. But for the purposes of V3, this summary takes you through all three components, looking at the AHI, which at the moment is the most widely accepted index for determining the severity of sleep-disordered breathing. So I said that I would come back to a handful of key questions. The first, way back at the beginning, was looking at the raw data dilemma. So what if the data on the tool are not available in its raw form in SI units? So in some tools, the sample level data are compressed into something like an activity count. So let's say we have an actigraphy device and it samples acceleration at, say, 32 Hertz. So 32 measurements of acceleration per second across each of the three axes. That sampling continues for the days, weeks, or months, however long the recording duration is. That's an enormous number of data points, especially if we're talking about a recording over many weeks. And in some cases, again, it's pretty common in some actigraphy tools, that volume of data is just too much for the memory capacity, for the battery capacity, and the ability to transfer data wirelessly, so using Bluetooth, for example. So in order to maximize the memory, battery, and data transfer capacity, the raw acceleration data are instead compressed into something like an activity count, which is no longer kind of meaningful for verification purposes because it's not in SI units. There is no ground truth for an activity count, and an activity count will differ across different manufacturers. There are different definitions. In that scenario, only the manufacturer can complete verification testing. There is no mechanism for someone other than the manufacturer to be able to take that tool and complete verification testing of the accelerometer against, for example, a shake table. So I mentioned earlier that V3 is considered a multidisciplinary collaborative process, and this is a good example. This is where a good dialogue and a good collaboration with a manufacturer of the device is key, because the manufacturer should be able to provide you with the results of their verification testing, summarizing the procedures that they adopted, as well as the performance threshold and their results. Question number 2, what is the best reference standard? So luckily in sleep, we have a very obvious reference standard for things like sleep efficiency and total sleep time and sleep staging, and that is an in-lab PSG. It is important to avoid comparisons to other estimated measures or previous generations of the same tool. So for example, if we have a tool, let's again use actigraphy as an example, and it is analytically validated against a PSG, but then now we have a second generation of that same tool. It's tempting to perform a comparison between generation 1 and generation 2 and show, ideally, that there's very little difference between the two. The problem with this back validation process is that if it happens over and over again, there is the opportunity for error to creep in and amplify over time. So by the time we get to generation 10, we may have data showing that it's similar to generation 9, which is similar to generation 8, et cetera, but it might be very, very different from the actual reference standard of PSG. So it is important to go back to the reference standard or the gold standard in each analytical validation study. It's a bit beyond the scope of today's talk, but I will just emphasize that the choice of a reference measure is not always straightforward. Again, I said earlier that sleep, it is, we have PSGs. I would argue that the manual scoring component of a PSG makes it suboptimal for use as a reference standard, but there are many other scenarios where there just may not even be a reference standard. So for example, if a digital tool is used for development of a novel outcome or a novel endpoint that isn't an estimate of an existing construct, then there may not be a reference standard in that scenario. DIME is actually in the early stages of undertaking a project on that topic, so more to come on that topic. Question 3, what if the algorithm isn't open source? I do hear pretty frequently calls for making algorithms open source. However, the reality is that many algorithms are proprietary and not accessible outside of the software developer. But analytical validation can be completed without algorithm transparency, as long as the investigator or the team responsible has an understanding of the input signals and the fact that they are verified. And the tool is able to give you granular data, so sleep stages at the epoch level or events detected event by event. Then the entire analytical validation process can proceed without needing to know the actual math behind the algorithm. So it isn't a requirement that the algorithms are open source, but what is critical is to have really strict version control. This is a problem or a potential problem for all digital tools, but it's a particular consideration for consumer-facing technology. If you are going to use an algorithm that is, developed primarily for consumers, the researcher or the investigator does need to be certain that every time there is an app update pushed to a smartphone, for example, that the algorithm isn't changing over time. If that is the case, then good version control will at least alert you to this issue. And one way around that, if possible, is to collect all of the sample level data, whatever it is that comes off the device, whether it's activity counts or whether it's actual raw data signals, bank all of that data up and then reanalyze it at the end of a clinical trial with a single algorithm, because that is one way of ensuring that all of your participants over the course of, say, a 4-year trial are analyzed uniformly. And then question 4 is, how granular should my analysis be? The trick here is to begin with the most granular analyses possible. So an epoch by epoch analysis or an event by event analysis, and only then move into a comparison of summary variables. So let's take, for an example, an auto scoring algorithm for PSG, which purports to estimate or label apneas and hypopneas throughout a recording. Now, let's say, hypothetically, your algorithm is completely broken and all it does is just label apneas and hypopneas completely at random. If the total number of apneas and hypopneas is the same or very close to the manually scored number of apneas and hypopneas, then your summary variables, like your total number, your AHI, that will be pretty much perfect. It's only if you go back and do an event by event analysis that you will uncover the problems with your broken algorithm. So that's what I mean by starting granular and then moving into analyses of summary variables. All right, I think I'm getting right to the end of my talk here, so I'm right on time. I will just point you towards some additional V3 resources, which might be helpful in your own work if you are considering using digital data collection tools in your studies and trials. First of all, about 6 to 8 weeks ago, I believe, DIME put together a searchable repository or library of analytical validation studies. The link is on the screen here. This is completely accessible. There's no need for a login or a membership in order to see the library and search within it. The library contains, at the moment, 208 manuscripts that focus on analytical validation studies across therapeutic areas. And 43 of those studies are analytical validation analyses of sleep-related outcomes. The paper that is listed on the screen here, this describes how we put the library together. I will just draw your attention to the fact that at the moment it's a preprint, so it is, you can read it, but it has not yet progressed through peer review. Hopefully that's not too far away. And importantly, when you are looking through the library, just be aware that we did not make any kind of judgment or assessment of the results themselves, so the quality of the algorithms. But what we did do is only include manuscripts that reached a certain level of reporting quality. And we made that assessment using the evidence checklist. So I'm sure many of you are familiar with the consort checklist, for example, for when you are writing or reviewing or publishing a manuscript summarizing a randomized trial. There is also the PRISMA checklist for meta-analyses. The evidence checklist is a publication checklist specifically for studies that fit within the V3 framework for connected sensor technologies. So I do encourage you to check that out if you are writing a paper or peer reviewing a paper on one of these topics. Finally, we have recently assembled a pre-competitive collaboration, pulling together a large group of project partners to extend the V3 framework in order to address human factors and usability. So when we wrote the original V3 framework back in 2019, 2020, we decided at the time that human factors, human-centered design, and usability were beyond the scope of V3. But we always had plans to come back to it at some stage, and that time is now. So the project partners you see on the screen have come together, as well as a group of academics, plus representatives of government organizations from multiple worldwide regions, patient representatives and patient advocacy groups, to extend the V3 framework by adding a 4th component specifically looking at usability. So we anticipate resources being available early 2024. So I will wrap up and end by acknowledging my co-authors who were listed on the screen here and contributed to the papers that I went into more depth on. The National Sleep Research Resource at sleepdata.org for making the MESA and sleep heart health datasets available to us, summarized in the 2021 JCSM paper. And finally to Philips for giving me permission to use some additional figures that were not included in the 2021 paper, but have been presented previously before now. So thank you very much. And I think we have at least 10 minutes or so for questions.